Thank you everyone for being here for our last seminar of this quarter. It's a great pleasure today to introduce Professor Rama Ranganathan, who's been doing some amazing work on proteins, and I've been hearing bits and pieces about this from some of the fellows who work at the Institute, so I'm really excited to look at what you've been working on.
Terrific. This is a brand new seminar. I don't think I've been in this room. This is new, right? Okay, very nice. Thanks for the invitation. I think it'll be really fun to engage with all of you on this problem. So I'm going to tell you today about this problem of the evolutionary design of proteins, but we're going to talk about proteins. And it's interesting because proteins are not the main focus of what I'm interested in, even though I've worked on these things for a long time. The more general problem, the reason I came to biology at all is described here. I mean, we all understand that biology is made up of these large dynamical systems that operate at scales from atoms to ecosystems. And it's been a major goal for all of us to understand their design. And don't forget that because these things came through an evolutionary process, we need to understand their origin through the process of evolution. It has been very difficult because the architecture of these systems, commonly is described as so-called complex, whatever this means. So the statement is that if you look at, for example, core metabolism in bacteria, and this is a portion of it. In some sense, this kind of graph or a picture is a triumph of human progress. 70 years of work, and we have found a majority of the parts and reactions that make up these systems. The basic questions, right, about what is being computed here. If I put an input into the system, what are the machinery? How does the machinery respond and produce outputs? Is not obvious. It's not just because there's a lot of parts and connections. There's a lot of strong nonlinearities embedded in the system, distributed in ways that we don't understand. So this has been just sort of the statement that it doesn't. I show you here at the cellular level, but if you look at every scale of biology, it looks something like this. And so what I will say is there's an approach which is really working, I think, to try to understand these systems in a more principled way. And I think about this as the statistics before physics approach, or the statistical approach. And the basic idea here is that, we've been collecting data across these scales of biology for a long time, and that's actually ramped up to be quite impressive these days, right? So the question for a lot of us is to say, can we compress this data into some representation, which comprise models that really can give us the design principles or rules of understanding and design, right? And I want to emphasize the change in the sort of shift in perspective, which may be obvious to you all today, but it wasn't obvious a couple of decades ago, which is rather than approach this problem by doing direct physical studies of individual instances of proteins or cells and so on, the idea is to take a statistical rather than mechanism-based approach to kind of find the effective dimensions of these systems and find the effective variables, I would say, and then enter a process where we try to understand the machinery. So this statistics before physics, as I say, approach is really actually changing our approach to biology. And I'm going to tell you that story from the point of view of proteins, because again, this is where most of the experimental power and past work has been done, so we can be more clear about what this kind of approach brings to us at the scale of proteins. So what are proteins? You all probably know that these are large molecules comprised of tens of thousands of atoms in principle. But all these proteins do the same things. They fold into the characteristic structures. They carry out function, and function is always some non-inclusive combination of binding and catalysis and allostery, the long-range transfer of information. And don't forget, again, because these are products of evolution, built in here is the capacity to evolve, right? All these features arise from information content encoded in the sequence, and that's an instance of our problem, right? What is that encoding, and how is information stored that delivers these properties? So let's take the statistics approach, right? This again started 25 years ago. The basic idea in this approach was say that if you want to understand this object on the left, you shouldn't go and do direct physical measurements on an instance of this molecule and try to understand the pattern of forces that deliver all these properties. Instead, pose the problem statistically. Go and collect yourself a large and diverse collection of sequences with one principle that they shared a common ancestor, that is, they're homologous, and with the principle that common descent implies common basic machinery, which was, again, 25 years ago, not an obvious statement. The idea was to look at statistics of amino acid outcomes at positions in the protein. And the idea was from this, would we get models for understanding and designing molecules? So let's just walk you through kind of an approach, right? So this approach called statistical coupling analysis takes it's kind of the idea is to try to understand the statistical structure of so-called conservation. So the idea is that if I look at this ensemble of sequences, I look at one position, like position I. So was there actually a stick or not a stick? There's no stick. Okay. I'm going to describe it to you then. If you look at position I, you might see amino acids that look like that, 50% phenylalanine. And the mean expectation of phenylalanine in all sequences in nature is probably more like 4%, right? So intuitively, we would say, look, an outcome of 50% given large enough sampling is really improbable. And that improbability is a statement of the evolutionary pressure at that position to be that amino acid. And this is simply expressed in mathematically saying that you can write down a measure of conservation, which is just the probability to see that frequency F given a reference expectation Q. And you all know with the simplest assumptions, this boils down to the Kullback-Leibler divergence, right, of the outcome at that position. And it's a way of like quantitatively saying what is the pressure on amino acid positions. Now, if you actually look at this in proteins, what does it teach you about proteins, right? So here I've done this calculation, this KL entropy at positions for a large alignment of these molecules called dihydrofolate reductase. This is actually a slice through the core of the protein. And I've simply colored the top 50%, conserved things in red. Everything else is in white. And you can see the basic result that's intuitive to all of us. The active site of the protein and the inside of the protein, so-called the core, is more conserved and the surface is less conserved. This is the kind of property that we've all know for forever, right? It's a well-known result. The interesting thing, though, is to go to the second order. So the idea is that where amino acids interact, the interactions are obviously important to deliver their biological properties. So the idea is let's take this to the second order. Again, instead of looking directly at frequencies and joint frequencies of amino acids at pairs of positions, in this approach, the idea is look at the correlation in the KL entropy, right, of these positions, which actually puts the conservation score, by the way, on a nonlinear scale, right, relative to frequencies. So if you compute a correlation matrix, again, this is not a simple covariance matrix, but if you look at the correlation in this information theoretic quantity, with, again, you can sort of analytically show that this produces a kind of coevolution matrix, which is a weighted covariance matrix. So the inside of this bracket is what you all recognize as standard correlation, right? It's the joint frequency of amino acids A and B at positions I and J, subtracted from the product of the marginals, which is which tells you how correlated these two amino acids at positions are. But then these are subject to weights. You can think about these, as our colleague Michael Brenner has explained, you can think about them as sort of Bayesian priors on your correlations. And these priors have a certain form, right? And so the bottom line is this kind of emphasizes the ancient correlations or the conserved correlations and de-emphasizes correlations that are due to local clades, right, that are more recent, with the idea that those are less likely to be physically significant. So that's a particular form of this kind of a statistical approach. This is what you get in the end, right? So if I take a particular protein family, like the S1A serine proteases. And remember that we're actually want to make statements ultimately about individual proteins, but this is an ensemble kind of analysis. Let's take all these sequences. So there's thousands and actually nowadays hundreds of thousands of these molecules in the database. You can make yourself this analysis, produce a matrix of coevolution that looks like that. So again, to be clear, what this matrix shows you is how every position of the protein going from beginning to end is correlated in evolution. Okay, these are not physical interactions, but statistical interactions, interacting with every other position going from N terminus to C terminus, beginning to end. Diagonally symmetric matrix, the diagonal tells you something about the intrinsic pressure on positions to be to have that outcome. And the off-diagonal terms tell you how much this position, the protein, talks to this other position in the protein, right? You get the basic idea.
Can I ask a basic question?
Yeah.
Sorry. So first, the marginal distributions that you compute the KL divergence with respect to, are they defined over the whole genome? Or are they defined like on a subset of the proteome?
Great. So he's asking this reference expectation, where do you get it from? So it's computed over the entire non-redundant database, all genomes, all proteins, right? With the idea that comprises the reference distribution of things that plausibly are proteins, right? It turns out that we used to worry whether that was going to be a very important thing. Actually, it's not, because it turns out if you compute it for a particular genome, you get the same reference distribution. If you compute it for a single family, you get the same reference distribution. Even more shockingly, if you have a long enough protein, if you compute it for one protein, it's the same. So it's just we stopped worrying about that some time ago. Although it is something that one should remember that is fixed in this thing. Okay.
So sorry. So the second order correlation you introduced, is that enjoy some sort of relationship with like a conditional mutual information between?
Yeah, it's like it. It's again, it's because it's phrased in this form of the correlations in the scale divergence. But conceptually, yes, right. But then we can look at the mathematics and see how they are in detail different. But the question, of course, was like, okay, here's a view through the lens of evolution, how all the amino acids are talking to each other in this protein. What does it teach us, right? So, I want to point out the challenge of analyzing the patterns that are here. The goal is to separate the the physical constraints, the evolutionary constraints, from these two sources of noise, right? There's finite sampling noise. I mean, you only have, 10,000 sequences. You're going to get a lot of spurious correlations just because of finite sampling. That's one issue. The second, more kind of complicated thing is historical noise, which I would say are pure correlations that come from the fact that sequences are not independent draws from some space, right? They're related by phylogeny, and the phylogeny could have functional meaning or could not, right? It could just be purely historical noise. You need to separate these things. So just to explain to you the process, maybe you all know what the story, but I'll just give you the analogy from finance. And this is not actually how we figured out this whole thing, but this turns out to be a good way to explain it. In the world of finance, people also solved a similar problem, which is to say, what is a good investment strategy in the stock market? We all know the basic idea is to put your money in as many independent modes of the market as possible. And as long as the economy is going up, you will, on average, make money. And the approach was that instead of paying your stockbroker to tell you where to put your money, you can do this statistically. So here are stock returns over some period of time for three companies, and you guys may recognize them. It's Chevron, ExxonMobil, and Citicorp, right? And it's sort of intuitively obvious that Chevron and ExxonMobil, being in the same kind of industry, are correlated, right? And the Citicorp is doing something different. And so the idea in this kind of approach was to say, well, let's make a covariance matrix over all the stock returns over this finite sampling of time, right? And then we can try to decompose this matrix and ask what are the modes, right, of variation in the market. And this problem has a similar issue that we have. First of all, there's a finite sampling of data, so you're going to be experiencing the finite sampling noise, same as the sequences. And then the point of the history, I'll tell you in 1 second. The bottom line is this is the eigenvalue spectrum of the covariance matrix of the stock market. These white things are the actual data, and then this black is random matrix expectations. The idea here was to say, if we scramble the time series randomly for every stock and then compute a covariance matrix thousands of times, you get a sense of what eigenvalues could be had just because of finite sampling, right? And indeed, that's what this shows. And so the idea was there's a bunch of actual eigenvalues that stick out from this, and we should interpret those, right? And the interesting story there is that you can break down the economy into the business sectors, right, by just doing this. And indeed, some much more fancy version of this is sort of under the hood in many of these sort of high frequency trading kind of approaches to the market. One thing I will point out is there's one gigantic eigenvalue always, which is actually the global economy going up and down. That actually coherently correlates all the stocks, right, going up and down. And then this thing then reflects simply the market cap of the company in the market. How much money do you have in the market? You get a large eigenmode. And that's interesting because that's actually sort of like history, what it does to sequences, because historical correlations care nothing about positions in a protein, right? They'll globally coherently correlate all the positions. So that has a similar concept as this idea of the global economy. So indeed, if you take so this is not stocks in the market. These are amino acid positions in a protein family. If you now carry out the eigenvalue spectrum of this, it looks like this. And the red is, again, the random matrix expectation by randomizing the alignment vertically at every position, right, and computing the eigenvalue spectrum that you could get. And so you can see that a large number of eigenmodes are basically consistent with just finite sampling noise. Now, I want to be clear, that doesn't mean that is noise. It means you can't tell, right? And I'll come back to that point in 1 second. But there are some eigenmodes that stick out. And then there's this large mode out here, which actually turns out to be dominated by these global correlations due to history. Interesting, right? So that's a way to kind of analyze. So the question is, in analogy to business sectors, what are these things that are significant eigenmodes of this thing? And it's quite fascinating to find out that if you look at the corresponding eigenvectors and actually kind of a manipulation of the eigenvectors to make them as maximally statistically independent as possible, all right, you find results like this where the vast majority of positions are not contributing to these top eigenmodes. And then there are like these, in this particular family, 3 groups of amino acids that stick out in a kind of quasi-orthonormal way. So these are like the sectors in the in business sectors, like in modes of the evolution of this protein. Sorry, there were some questions. Aditya.
A question about the biggest eigenvalue.
Yeah.
Is it not coming from just an overall mean signal? Like if you just subtracted off the means, so everything was mean zero, would you get?
Yeah. So one way to actually that top mode is the average correlation, right? And so you could that is what it is actually, right? But it turns out to be dominated, right, from a process that globally coherents all the stocks. It's so absolutely true that top mode cannot be thought of as like just noise and you can just clean it, right? It's because actually it carries also very important functional information, but the whatever process globally correlates positions will show up there. Yeah, will be dominated there. Was there something else?
Yeah, we have a question from online. It's from Madav. Has anyone done subsampling with bootstrapping of the MSA and studied the trends?
Yeah. Well, first of all, I will say that the way to derive the analytical form of SCA is basically a bootstrap approach to looking at the correlations in the entropy function. And so I'll be happy for anyone. I'll send you a little short 2-page note that just explains to you the mathematics of that. So it really is a bootstrap concept to look at the correlations of the entropy function of positions, right? But if you do bias sampling from the alignment, then all kinds of weird stuff happens, obviously, right? So that's let's not talk about that right now. It can be interesting, but it's not something we need to pursue right now. Okay, so an analogy to the business sectors, we call these things protein sectors. So that was the idea of modes, evolutionary modes, collective modes in the protein. And a really fascinating result was that there was no imposition on this method that these things needed to be physically connected in the protein structure or anything like that. But indeed, it turns out that every one of these collective modes in evolution are some physical network in the protein structure that actually made some sense. So the green network is actually the core catalytic machinery of this enzyme. It's the famous His-Asp catalytic triad that does hydrolysis of the peptide bond in this case, and a bunch of associated residues sitting at the interface of these 2 beta barrels that make up the protein active site. The red network was also interpretable. This thing was built around the primary specificity determinant in the substrates of this enzyme. So these proteins are specific for cleaving after some amino acid, and indeed this whole network was historically understood as being the thing that controls substrate specificity in this thing. And then the blue thing was not obvious what it was, but we did experiments on this stuff. One of the interesting results that came out of this was really that these orthogonal, quasi-orthogonal evolutionary modes correspond to quasi-orthogonal biochemical modes in an individual protein where the experiments were done. So the data here show you mutations in the red network in red mutations in the blue network in blue. And what's plotted here is the change relative to wild type of the catalytic power of the enzyme. And on the y-axis is the thermal stability of this protein, right? And the result is that you mutate the red network, and yeah, sure enough, you affect catalytic power quite a lot, but you don't really touch thermal stability. And then you mutate the blue thing, and you significantly affect thermal stability, but you barely touch the reaction coordinate of the enzyme. And this was really surprising because nobody ever saw these sort of independent modes of variation that it's like nature has these control knobs, right? To be able to steer the molecules and these different biochemical properties embedded in the evolutionary correlations, right? So the sectors turn out to be biochemically independent elements within proteins. I'll make a point about how non-trivial this is. We all, those of you that learned any protein biochemistry in class, you learned the traditional hierarchy of protein structure. Primary structure, which is the sequence, the linear sequence. Secondary structure elements, beta strands, alpha helices, loops, this kind of things. And then the tertiary structure, which is the folded structure of the molecule. And then there's quaternary structure between molecules. It turns out sectors don't follow any of this hierarchy. There are some different descriptor of the properties of proteins. So you can see in the top panel that the red, green, and blue sectors come from all over in the primary structure. They have nothing to do with primary structure. It's not obvious they have anything to do with secondary structure. And if you look in the 3D structure, these are all networks that are embedded in the core of the protein. They even share interfaces with each other, physical interfaces, but apparently mutating one does one thing and mutating the other does something else. That kind of mechanical decomposition of the inside of the protein is not at all obvious when you look at proteins like their 3D jigsaw puzzles, with this sort of homogeneous pattern of contacts inside, right? So this really opened up the idea that, first of all, sectors are a new language for proteins, right? It's a new descriptor that actually corresponds to the evolutionary units of functional pressures. And it spoke dramatically to a kind of mechanical, decomposability of the forces inside of a protein to be orthogonally represented like this, right? And that was not obvious. I will say, however, don't forget that proteins are like marginally stable objects that sit with the free energy that's near zero, right? And there's a lot of interatomic interactions that are in there that are basically have values that could be right around zero. So it's not at all inconsistent with a 3D folded protein that there could be that kind of mechanical decomposability in the protein. It's just that nobody ever saw it before, right? So that's the story. So I want to just say, sectors in some sense are asking for a new paradigm, a new language to speak of proteins in terms of these evolutionary units. So there's been just a ton of work in the past on understanding what these features of proteins do for folding, for various aspects of function, including allosteric signal propagation and all that. And then more recently, the role in evolvability. But I'm not going to tell you about any of this stuff. These are references. You can look them up to if you're interested in these features. But I want to focus on two aspects, actually. And one is about the spectral decomposition and what it really means and how can we test it experimentally? Because, this is a statistical analysis of evolution. What does it have to do with the physics of individual proteins, right? We should really need to nail that kind of relationship. That's always been a supposition that these physical networks in the protein tell us something about the physics of proteins. Can we get at that? So I want to just talk about that for a few minutes. So, okay, first of all, I told you global coherent mode, these significant collective modes, which are these sectors. What about all this stuff underneath the distribution of eigenvalues that are simply consistent with finite sampling noise? As I said, the data don't say these are irrelevant, just that you can't tell, right? It actually turns out that they are really relevant, and let me show you how. So another kind of approach to analyzing the same data is to make these sort of probability distributions for proteins. So in one approach, this is the approach of making Potts models. The idea is, again, you extract the same statistical quantities, frequencies and joint frequencies. But now what you do is you make a proposal that the probability to see any sequence sigma is a Boltzmann distribution of a Potts model, right? And then the Potts model here is parameterized by fields, constraints on amino acids at positions, and pairwise couplings between them. And of course, in the way that you do this kind of model, you infer parameters H and J with a maximum entropy constraint to satisfy the empirical frequencies and joint frequencies, right? Okay, one can do that. And I think about this as kind of another way to represent the information content in the pairwise correlations, right? But it has an interesting difference, which is not really obvious at all to people, and I want to explain that to you all. One of the features of this inference is it's always done in the limit of hideous sampling, right? I mean, you have like this many sequences, and you're trying to learn like that many parameters, right? So this is a horribly undersampled problem, right? And to keep parameters from exploding to infinity, we all impose some kind of regularization. Typically, what's done here is an L2 regularization with and how do you set the regularization parameter? It's typically just set to be something small so the thing doesn't blow up, right? In the inference, which is typically on the order of 1 over m, where m is the number of sequences you have, right? So if you had 1,000 sequences, you'd make it like 10 to the minus 3 or whatever. It just keeps it on the order of 1 over m. There's a rationale for the 1 over m, by the way, sort of the natural temperature, some sense of the alignment. But here's the story. It turns out, and I don't have the time to tell you this in the talk today, but you can look at it in this paper, which is really important, I think. It turns out that without corrections, the Potts model with the standard regularization people use, the small regularization, it really sees only the weak eigenmodes of the correlation matrix. And one simple way to think about this is the mean field solution to the inverse problem is to invert the correlation matrix, right? J is like C inverse. And so the top eigenmodes of J are like the bottom eigenmodes of C. And so, it's basically seeing these weak, all this stuff. And actually you can show, as this paper shows, this is Yakov's work, that you really suppress these collective modes in the inference and it's fundamentally a problem of inference with undersampled data, right? So if you had infinity sampling, this would not be a problem. This is a problem of undersampling and the influence of noise due to undersampling on inferring these features, okay? And so the bottom line is what you learn from the bottom eigenmodes is the contact graph of the protein. This, of course, is the work that launched a whole line of work in structure prediction, right, with sequences, because you can so the thing on the right is the empirical contact graph of this particular protein, an SH3 domain, gotten from looking at many crystal structures, okay? This thing is the top modes of JIJ, right? And actually, you can really get the contact graph of the protein with enough constraints that you can fold that protein without any physics or any kind of other information, right? And that was shown by our colleagues Debbie Marks and Lucy Colwell and so on 10 years ago, and then dramatically amplified by Google and so on. And now with models like AlphaFold, the idea is that in part it has the soul of it is in here, right? The idea that you can get the contact graph from these weak eigenmodes, right? So like the spectral decomposition breaks down this structure into a kind of pattern like this, where there's a global coherent mode that that we've already talked about. It has distributed collective modes that correspond to these functional elements in the protein, right? And then there is a bunch of these local contacts in the structure surrounding the whole, that making up the rest of the structure that represent the local environments in the structure, right? So the basic statement I would take from this is that it breaks the 3D structure. It says that proteins are really heterogeneous materials with like high order epistatic constraints, that is high order nonlinearities in these distributed collective modes, collective features, surrounded by a sea of weak local couplings, low order epistases like pairwise or even independent, right, kind of positions. And I think that demands a test. I mean, this is a statement that this organic crystal-like thing that you get to see in a crystal structure is actually a heterogeneous material with like collective modes embedded in a sea of weak couplings, and that needs a direct experimental test. Yeah.
where you're leading, but can you give us some like counterfactuals of like a no model? Like
Yeah.
how much of this is due to the factor of proteins that are evolved quantities versus like if you took a random stream of amino acids?
I think it has everything to do with that, actually. So this is the question. There's a beautiful problem, which is what is the origin of this pattern, right? And that's the line of work that I'm not going to talk about today, which is about the evolvability, right? It turns out that if you evolve proteins, and this is you guys know Eric Rivera probably. So Eric's whole thesis was about trying to understand the origin of this kind of architecture in just with simple toy models, right? And we also have a lot of experimental data on proteins. It turns out that when you evolve these sort of frustrated networks under conditions where the environments are fluctuating, right, and they have to be adaptive to changes at various timescales, that you end up with these sort of patterns or natural outcomes of this, right? Very important. Different line of work.
Converse is also true. You're saying that like
Yeah.
if you just have random in biophysics, but like this different structure.
Totally. So I think one of the really important concepts here is that there are many ways to make a protein. This is not the only way, right? So indeed, you could make an optimally packed protein. I think our colleagues in the protein design community have shown this up and down, right? That you can repack a protein core to optimize all the local interactions to maximize the van der Waals force, and you get rocks, right? You get proteins that you can boil and they don't unfold, right? You get really stable proteins. And so there's a long line of work that shows that proteins are not optimally packed in the core, right? Actually, if you look at the distribution of free volumes inside of a protein, it doesn't look like a solid at all. It looks like a random collection of nuts and bolts in a jar. That is to say, you get a very broad distribution of free volumes in a protein. And so people have known for a long time that proteins are actually not looking like just solids or something like that, or crystalline materials. So I think that there's a lot of data that argues there are many ways to make a protein. This is a way that evolution prefers for reasons that Eric and others have begun to expose, right? All right, so I want to put this to a test because this just needs a test, right? And this is a crazy heroic experiment that was done in the lab. I want to describe it to you. The idea was this. Let's take a protein, that thing called the PDZ domain. There are 83 positions that you can align in the whole family. And the idea was to mutate every position to the next most common amino acid in the alignment. Do you get what that is? That's like attempt to try to make a subtle perturbation at every position, right? And every one of those mutant proteins was isotopically labeled with N15 and C13 to be able to measure the chemical shift of every backbone amide proton, nitrogen, and carbonyl carbon. Let me explain what all this means. The protein is a string of amino acids connected by peptide bonds. By doing this experiment, you get to see the chemical shift of this amide nitrogen, this amide proton, and this carbonyl carbon, which is like a signature of the local structural feature property around those atoms, these nuclei, actually, right? So what I can do in this experiment is I can mutate a protein at one place and look at the effect, right, at every peptide bond throughout the entire protein. And this is like the most sensitive measure of structural effect, right? So it's a really good measure. And the idea is that we collected that data. By the way, for technical people here, that's called an HNCO, chemical shift experiment. And it was done, the entire data were collected with and without the ligand too. So we wanted to see what the ligand does. So completely crazy data collection, but this is what the data look like, right? So look, this is on the left, this is all the 83 mutations going from beginning to end, ordered by the primary structure. And the rows are all the amide protons, amide nitrogens, and carbonyl carbon chemical shift. This is the change relative to wild type. So white means it's same as wild type. So you can see there's some pattern, right, of perturbation. That's without ligand. Everything was collected again with ligand, okay? And the question is, how do you analyze these data? And you guys know what to do, but I'll just show you some so cherry-picked examples. If I take two columns here that are far apart in the primary structure, and I look at the chemical shifts, so the red is the site of mutation, blue is the largest effect, the white is the next shell below, and after that it's not statistically significant, so it's not shown, okay? Right? You can see the story here. These two positions are packing neighbors in the structure, and they show kind of a propagated and similar pattern of chemical shift change in the structure, right? So this is a kind of examples of things that show kind of propagated and some, I would say, correlated effects. But then check this out. There are two other positions I can just pick that are also far apart in the primary structure, that are also packing neighbors in the 3D structure, but they have completely local and non-overlapping effects. These are idiosyncratic local effects, right, in the thing. So I'm just giving you a preview that these sort of different features of this. Of course, the whole data looks like that, right? It's like every position of the protein, and you have this thing. How do you analyze this data? I mean, again, the obvious thing to do is to compute a covariance matrix of all the columns of this data, which represent how all the mutations look like each other or not, right? Make an eigenvalue spectrum. The red line again is the random matrix expectation. Proteins are finite sized networks. You can like look at what eigenvalues you could get by just finite size of the protein. And indeed, you get the idea that the vast number of eigenmodes again are just consistent with the finite sized network. I mean, sort of idiosyncratic local sort of effects. And then there are some collective things, right? You can back compute the data corresponding to the weak modes and the top modes, right? And this is what you get, right? So anybody can guess what that is? What is that? Well, it's the contact graph for the protein, right? So the thing on the left is calculated from a structure of that domain. This is the perturbation, the weak eigenmodes of the perturbation analysis. So this is like what you should expect, right? If I go around making a perturbation at every position in the protein, and I should at least see in the weak eigenmodes all the local contacts, right? Okay, so indeed you get the contact graph back. So again, it tells you that the contact graph lives in the weak eigenmodes of this perturbation analysis. What is this thing? The top modes contain some other pattern, and what that is in this case is a mode that connects this loop of this protein through this internal alpha-1 helix to the backside of the protein, this alpha-1 beta-4. This is a kind of a collective unit, apparently mechanically in this protein, which is in these top eigenmodes. Aditya.
So why are these matrices not square?
Oh, yeah. Sorry. These are just truncated because you have a set of positions you mutated, right? And then you read them out though at all the positions in the protein. So the number of mutated positions is smaller than the number of readout positions.
Okay, so you didn't mutate every single.
Well, we mutated every alignable position in the thing because we were also trying to compare this to, as you'll see in a moment, to the evolutionary modes, right? So the idea was, that was the idea. Okay, it covers the entire core domain basically. A lot of these proteins come with extra modular components, right, which you can use for the readout, but they are not actually part of the core domain, right? Okay, so the top mode looks like this, but this is in the absence of ligand. This is the APO data set, so to speak, without ligand. What does this whole thing look like with the ligand? Well, now you get this, right? So again, same thing, you get the eigenvalue spectrum. The weak modes contain the contact graph. The top mode, so the weak modes, by the way, barely change between and you can say this quantitatively, you can barely change between the with and without ligand. So it's like saying that most of the protein doesn't see this ligand at all, but this thing really changes, right? It really remaps the top modes. And now the top mode is this network that connects this beta-2, beta-3 loop, alpha-2 helix, this beta-1, beta-2, alpha-1, alpha-1, beta-4. It's this kind of extended thing. Well, I mean, what I can tell you is this tells you that there's a mechanical collective mode in this protein. And what's, of course, interesting is that this is actually nearly exactly this evolutionary collective mode that you get from sector analysis in SCA. And by the way, I'll make one point. You might notice that there's in this beta-2, beta-3 loop, there's a bunch of extra atoms that show up in this mechanical unit. And in the carboxylate binding loop, you get a bunch of atoms over there that show up in the mechanical unit. There are data that we have that show that these amino acids, that one and this one here, which are glycines, actually control completely the conformation of those two loops, actually, right? So these are like units that are mechanically just sort of slaved into the configuration of these. You guys might know glycines have special backbone characteristics that can restrain their geometry in a way that's different from other amino acids. So the bottom line is individual proteins indeed have collective mechanical modes that engage the ligand, because this is what I showed you, right? And that correspond to these sectors. I think that basically gives us a experimental view that this sort of basic pattern of having mechanical collective modes embedded in a sea of weak couplings is indeed actually consistent with experiment. So I want to say that what I conclude from this is that proteins contain vastly different scales of cooperativity inside them, epistasis if you want. A few collective modes surrounded by a sea of weak local couplings, and these collective modes encode the reaction coordinate, right? The evolutionary conserved reaction coordinate. But yes.
So it seems that perturbation really highlights that evolutionary lynch. If I wanted to work backwards, let's say that I have a protein and I want to understand what is the evolutionary dynamic.
Yeah, you could do the experiment, you mean?
Yeah.
Totally. Insane experiment. But that's been one of the limitations, actually, right? We've never had easy experiments to visualize these mechanical units in proteins, right? So I agree with you. I think one should be able to get sectors out of it, experiments on individual proteins. It's partly why we set up this whole electric field crystallography thing at Argonne, was a way of trying to just put forces into proteins and excite these collective modes and just see them. But that's another crazy technology that took 10 years to develop. So I'm just showing you things that this is what's limited our understanding of proteins, is the inaccessibility to this kind of data, right? I think it's kind of changing now, so it's good. But you know, it's an important statement that it's non-trivial that which evolution is preserving over time and speciating over different like individuals of a protein family can be learned, right, from that data set. And it's not that every protein is making up its own configuration of forces to do whatever it's doing in whatever eco niche it's operating in. If that were true, then none of this would have been true, right? So that's a not trivial statement. Okay, I want to. The second point I want to make, besides the connection between evolution and mechanics, which is for me a long fought battle, okay, is this thing, that these are stupid low-dimensional simple models. I mean, you know, here we're just extracting first and second order statistics. We're ignoring any higher order terms, and for that matter, we're fighting huge amount of noise, and even the pairwise correlations are being learned when there's highly undersampled limit. Aditya, please.
Just going back to the previous thing, I guess you showed that there's one of these sectors which corresponds to the mechanical mode associated with binding to the ligand. So what do you think the other sectors are doing?
So like in the case of protease, right? One of them is actually about the chemical reaction, the K-cap, right? One of them is about substrate binding, which is like the KM, right, this thing, more like the specificity for the substrate. And the other one is actually controlling the lifetime of the molecule, okay? So I consider those, which means like the autocatalytic degradation rate or whatever, the lifetime of the thing, you can set it with this blue. So I just think this means that the fitness function lives on a multidimensional landscape, and sometimes that landscape can be orthogonalized like this. That orthogonal business is not, to me, fundamental. It's not guaranteed. Like I think that comes from the fact that the environment that these proteins evolved in actually fluctuates orthogonally, and therefore you get this result. If there was coherent or correlated fluctuations of, say, temperature and some substrate or something like this, I think these would not be separable features.
But in this experiment, you were able to look at the
binding
mode.
In this case,
with it without ligand.
Right. So in the PDZ, there's only one sector.
Oh, I see.
And there's a bunch of proteins in which there's only one sector, right?
Okay.
Which is another way of saying that if there is some multidimensional thing going on, it's all embedded in this one thing, right? So that's one of the reasons we chose it as an example for the experiment, right? But it would be fun to do this with the protease and see this mechanical independence of these things, right?
That's a question before you go on to this.
Yeah.
Is bothering me. I think you may have answered it already, but to what extent is the sampling problem we were discussing earlier a problem for data collection, and to what extent is it an evolutionary problem? In the sense that evolution hasn't sampled.
It's super. I'm going to show you a bit of data. Can you hold that thought? I'll come back to it because it's really cool. Great question. Insightful question. All right. So what I'm going to ask is, like, this is like just extracting some. So is it like tip of the iceberg kind of information, or is it like everything that nature knows how to make that protein, right? So that was a question we asked 20 years ago. And so the only way to answer that question is to put no knowledge about the structure, put no knowledge about physics, just generate synthetic sequences from the representation you have and ask in the lab if those are folded functional proteins or not, right? So just, can you make these proteins? And so indeed, I'm going to show you the data because this is ancient stuff. So I'll just tell it to you. On the left are examples of proteins that nature made. This is the human PIN1 WW domain. This is a rat brain PDZ domain. On the right are experimental structures, importantly, right, of synthetic proteins that came, again, with no prior knowledge of the structure or any physics, just satisfy these pattern of correlations, right? And with that kind of sequence identity, with that kind of probability, you can make folding proteins. And as you can see, these things are, quantitatively, the root mean square deviation of these structures to their natural counterparts is within the range of the natural proteins to each other, right? So at least at atomic resolution, you can make proteins with, it took 8,400 numbers to make a WW domain, okay? That's the actual data, right? I mean, now it's just funny to me that we've got these AI models with 98 billion parameters, and we're trying to, it was 8,000 numbers to make this protein, right? The important thing about this paper, it really argued, it set an upper bound on the complexity of the information stored in the sequence, right? Okay, it wasn't just folding; these things work the same as natural proteins. So WW domains are polyproline peptide binding modules. This particular natural one likes to bind PPXY-like motifs. Indeed, the synthetic one binds with the same specificity and affinity those class of peptides. Okay, so this is like, as far as we can tell, it's a perfect reproduction of what nature has done. Aditya.
So if I didn't even put in the pairwise thing, I just made
it. Look in the paper. Okay, nothing folds. Yeah. So the profile model, so to speak, with only first order statistics, is not even making a protein that can be. Typically, they're not even soluble. The few that are soluble are definitely not folded and not functional.
But I thought people say that if you just take consensus sequence.
That's a difference. So you realize, Aditya, the consensus sequence is a cryptic way of putting correlations. Because what you do is you pick the most prevalent amino acid at every position and make a sequence. So that's actually an important result, that the consensus sequence tends to be, it's kind of like the ground state of the Potts model or something. You say, it's like you pick and it's interesting because those sequences tend to be hyperstable. Like I got to overpack the information in that thing, right? Okay, anyway, just to finish this point, this synthetic PDZ domain binds one ligand in the mouse genome, which is in the same range as all the mouse domains that bind in that region. So basically, at least in vitro, these data said that you can get folding and function back with just a tiny amount of information, right? This is like, this has been expanded upon significantly in more modern era. So here's Potts models. So we set about trying to make this enzyme called corsmate mutase, which operates in this key branch point in this biosynthetic pathway that leads to the production of the aromatic amino acids tyrosine and phenylalanine. So bacteria, for example, that lack this enzyme are oxytrophic, as we say, for these amino acids. That means they can't grow if you don't give them these amino acids. That sets up a very beautiful assay for these things. So, there's we made again, 10 to the third, right? Natural sequences. If you take all the 10 to the third and you stick them into E. coli and you ask, do they work? So this is a measure of the ability of the protein to work in vivo, where 1 means you work like the E. coli enzyme, 0 means you're dead, right? Okay. The bottom line is that 40% or so what nature makes works in E. coli, okay? By the way, why is it not 100%? It's because there's some context dependence of these proteins, right? Like not all the corsmate mutases you take from every organism will work in a particular one. But this sets the gold standard, I would say, for a model that could generate synthetic sequences. So indeed, you make a Potts model from this thing, again, only using pairwise statistics, sample from the Potts model, and with that kind of top hit sequence identity, you can reproduce the distribution you get in nature. So Potts models can make synthetic enzymes that are basically indistinguishable from the natural counterparts. Of course, we also purified a few of these and measured all the kinetic properties. They're all exactly the same enzyme, right? With dramatic sequence variation. one amusing thing about the Potts model was it allowed us to compute the entropy of the sequence space, right? You can compute the entropy of a Potts model. So you can say how many sequences are consistent with that, right? A thing. And the latest estimates from Emily in our lab is that it's lower bounded by this number, okay? And 10 to the 54. So it's like a completely stupid number, right? It's a gigantic space in which evolution apparently is sampling. So that what I'm saying is you can go to that space and take 1,000 sequences and 40% will work in E. coli, right? Okay, but the amusing thing about this number is that it also happens to be zero, right? In the sense that without constraints, you have that space that you're looking at. So you can never find the space in that space without the correlations. This goes to your question. If you look at the profile model, it's not a good representation of the space, right? But once you have the data to make a model like that, you can then play in this gigantic space of alternative. I think about the spaces like alternative histories that could have happened in, I mean, evolution is a stochastic trajectory. If you went back and reran it with exactly the same environmental history, you'd be sampling from the space. It's basically the idea.
What does it mean for a sequence to be consistent with the model? It's like with
basically that the probability is that the energy spectrum is in the range of the natural proteins. Yeah, exactly. Yes.
I mean, it feels like that space is still too big. I see that.
Which one? 10 to the 54?
Yeah.
So that tells you how simple the constraints are, right?
Yeah.
The fewer the constraints, the bigger the space. So it again goes to what I was saying before. You don't need that many numbers to make the protein. And indeed, this number reflects that. Yeah. I mean,
comparing it to your point about regularization?
Yeah. So one of this number, when it was published in that paper, it said 10 to the 25th. It went up 20 log orders because we fixed the regularization problem. And this is Emily's thesis. You should talk to her about it. Yeah. So it's amusing. The space expanded 20 log orders, and the fraction functional sequences didn't change, basically, right? So if that doesn't convince you that these models are overfit, right? I don't know what will, right? So so actually my point, there's a cottage industry of people that are making proteins from these kind of sequence-based models and trying to like commercialize and do all these things. But I just want to point out that all these things are totally overfit, right? Indeed, you can see the overfitting just here, right? So the bottom line is that if you look at natural proteins, what I'm plotting here is the function on the x-axis, sequence identity to the nearest natural protein on the x-axis, right? The y-axis is the function. 0 would be like wild-type activity, and this would be like dead. You can see that nature makes sequences with a broad diversity. I mean, by the way, if that graph doesn't convince you that sequence identity has nothing to do with anything in terms of function of the protein, I don't know also what will, right? Like you can get sequences that are both non-functional, functional throughout this range. But if you look at sampling from the model, it's really cut off, right? You can't make things that are like in this very diverse because these models are just really overfit. And again, it's because you are fitting the model to a ton of sampling noise in here. You're getting the contacts, and there's a ton of sampling noise being imposed, and so you're stuck, right?
What does natural non-functional mean?
It just, again, it's these sequences that are Corsway mutases, but they don't work in the assay in E. coli.
Oh, but they're from the MSA.
Oh yeah, they're just from the MSA.
So they don't work locally in that organism.
Right.
Got it.
Right.
Got it. Okay.
Right. Exactly.
They work somewhere else.
For sure.
Yeah.
For sure. Right. And so. So that's the story of like, I don't think the models don't really explore the space open to evolution. And Emily's whole thesis has been about trying to clean the model so we can actually see the boundaries of natural sequence space. And I encourage you to invite her to give a talk sometime because it's a beautiful story actually. So Aditya, sorry.
What does it mean to have natural proteins which are far in sequence identity from the nearest natural?
Just means that they're far from the nearest natural.
Aren't they themselves the nearest natural?
Oh, I mean, excluding the self identity.
So they're like so there are a bunch of natural things
sitting out there somewhere, really far from anything else in the database. Exactly. Yeah. So I mean, to be honest, if you really had a good model, you'd be able to just systematically make sequences in the top left corner of this thing, right? If you really understood that. So I'm going to try. So okay, By the way, if you want Emily's work, if you want another description of overfitting, here's a story. You sample from the POTS model, you get this result, right? I showed you already. You throw away 98% of the parameters in the POTS model, right? And she just throws away all the weak stuff. And that's the result if you generate from that thing, right? So if that doesn't convince you that these models are overfit, then I really don't know, right? Because this is what she did, and the entropy of this is 10 to the 25th, and the entropy of this is 10 to the 54, right? But with like no change in the. And so I think that's pretty amusing. Okay, let me just in the last few minutes just tell you about a new model because I think it actually be relevant to our discussion about noise. So this is one of these fancy models, right, in which we've integrated two language models, a protein language model that was trained on the protein universe, and a literature language model that was read all the papers. So it's a PubMed model, right? And then a joint embedding space was made with contrastive learning. The output of this space can then go through a diffusion transformer and make a synthetic protein. Vastly more complicated a thing than everything we made before, but it allows you to give a text prompt and make a protein now, right? So what's the value of that? So here's the kind of thing you can do with this thing. You can like write a piece of English, say, I want a protein that looks like this. That text gets embedded in this latent space somewhere. The decoder makes a protein that lives there. And this is an overlay of the synthetic protein in color and a natural protein that lives there by AlphaFold prediction, right? So with really good statistics, this thing is basically showing you can make different structures from different parts of this space, but albeit with text prompting now, right? But the real killer is function. So I just want to show this. This is the model system. There's a pathway in yeast called the SHO1 pathway, which is a stress response pathway that deals with osmotic stress. It involves a critical interaction of an SH3 domain with this target protein. This interaction is critical to mediate the high osmolarity response. So actually, if this doesn't work, these yeast will not grow in conditions of high osmolar strength, okay? And indeed, if you take 8,000 natural SH3 homologs and stick them all into the SHO1 receptor and ask if they work, the answer is like 2% of them work, right? There's like 176 of them, and all of those are orthologs of the SHO1 SH3 domain in all the diversity of fungi. This is not a trivial result because these fungi diverged like 1 billion years ago, right? Apparently, you can borrow an SH3 domain from some rust and stick it into Saccharomyces cerevisiae, and it works just beautifully, right? But there's only a tiny number of them, so that's a real test for a model. Can it do it? So let me just show you. You can write a piece of text. I want an SH3 domain that binds PBS2. It works in this osmolar pathway. It activates a MAP kinase signaling pathway. That's all the information you give it. You make sequences, you stick them in there, and this is the result, right? So the blue dots are all text-prompted proteins, right? The bottom line is you can make a bunch of proteins that are actually really as good as the natural protein with a text prompt. The interesting thing is the difference between the top graph and the bottom graph is the quantity of information you give in the text prompt. So if you put a lot of information in the text prompt, you make a lot of functional proteins, but the top hit sequence identity to the natural world is like 78%. But now if I ask a much more stripped down prompt with a lot less information, I make proteins that are vastly more diverse that still function, right? And going to Seppa's question is what I wanted to get to it, that this diversity is now beyond the diversity of all the natural proteins that work in that functional context, right? So I think in some sense, this starts to make me think that maybe like in some sense, evolution gets kind of stuck in its ability to generate diversity because of evolutionary dynamics. The actual physical constraints to do that job can be less than what you've seen by evolution. And this model somehow by this multimodality business, it's allowing you to explore some larger space even that nature has explored for this particular function, right?
So evolution's seen less of the 10 to the 54.
But than it could have. And if you went back and reread it, it probably would see that space. But because of the dynamics of evolution, it gets kind of stuck, right? Which is interesting. It has to make an answer to the question of why are sometimes orthologs so damn similar to each other when it's obvious they don't need to be, right? And I think it really is a deep problem of understanding how evolutionary dynamics puts constraints on top of physics, all right? Okay, I just want to make one last point that You can see, though, that the model, in some sense, doesn't know anything about function because it doesn't know how to dial in sequences that live in the red box, right? That is, say, things that are actually just functional. And what I'm saying is the model is making a bunch of dead things. If it really understood function, it would just make things that are functional. And I just want to leave you with the fact that what the model is missing is collective modes. Going back to our whole discussion today, it turns out that if you compare the functional proteins and the non-functional proteins, that there's a network of 14 positions in this protein that become invariant in the functional group that are being mutated in the non-functional group. Those are precisely the sector in the SH3 domain, right? So if you dial in, if you condition the design on sectors being perfectly intact, you get a very large fraction of things that are functional. But if you mutate the sector even a tiny bit, like 2 or 3 mutations, you're dead because it's a collective mode, right? So what it first of all says is that there's a kludgy solution by just screening designs that have sectors intact, but it really begs the question of why the model isn't aware of this already, because it's trained on the sequence data. And you know the answer already, because I've told you the answer. The answer is there's different scales of epistasis, and the model it doesn't see these collective features properly, right? It's the way that model inference is done right now. It tends to emphasize these local structural contexts, very good for structure design and structure prediction, but not the path to actually getting function design, right? So I think that correcting this problem of inferring different scales of epistasis in the condition of undersampled data is like the unlock for this thing. And that, we got to do it. Because I want to end by simply saying that it has nothing to do with proteins. This problem is a general problem of any data that contains vastly different scales of nonlinearity in it, and you're trying to infer it with a data-driven model, you will experience this problem, right? So this is a problem that's been exposed at the protein level, but it's got nothing to do with proteins. So this deserves attention by anybody trying to make these days generative models for biology. So anyway, these are the wonderful people in the lab who are doing this work and have done this work. I want to just highlight Niksha, who did the BioM3 model, the last thing. Mike, Miranda, Emily, and Niksha all together have done all this recent work on CARS mutases and all that. And there's been a long history of people because I presented things from 20 years ago, so I don't put all their names here, but there's a giant group of people that have worked on this. And I want to thank the collaborators, particularly Andy Ferguson at UChicago, that's worked with me on these new generative models. So anyway, ask questions, please. Yeah. Yes.
Really amazing. I guess I want to ask if you'd hazard speculation in opposition to what you invited us to do at the beginning, which was to think about statistics instead of physics.
Yeah, correct.
So if you instead said, I'm going to coarse grain the alphabet of amino acids by a physical quantity or by a physical quantity like the hydrophobicity.
Yeah.
And there's a long history of these old alphabets, right? Like
yeah.
HP, whatever. And to first order, it's like, well, maybe like one amino acid shares 5 others, like a similar hydrophobicity value. And then you did sort of the game that you were talking about with function and like the sort of spread of of possible proteins that are consistent with a given like total folding energy. What would you get? And like, how do you ever like, is that part of the sort of later on game that you end up playing?
Yeah, I think those have, you're right. People have done like the sort of hydrophobicity patterning and all this. Michael Hecht, for example, has made a lot of coiled coils and all these, and even done functional proteins with that stuff. I don't think that's actually getting us any closer to understanding natural proteins, to be honest, because I don't think that the local chemical property of positions is the correct parameterization of the protein problem. For example, there are positions of proteins that want to be phenylalanine and lysine. It's not a mystery, right? Because in some proteins, that position is making packing interactions or pi-pi stacking interactions with something next door. In some other proteins, there's a salt bridge being made with this thing, right? So I think the invariance is the free energy the coupling free energies, that is the energetic landscape, is the invariance. I think there's a giant degeneracy of mechanism that can give you the same energy landscape. And that's the entire problem of going from physics to models, because you're working with specific instantiations of the molecular forces, and you start thinking like, oh, that salt bridge is really important, but it's not important because in some other protein, the thing is a disulfide bond or some packing interaction, right? So I think that the general, I would like to say that the approach of trying to coarse grain amino acid alphabets and use that as a way to lower the dimensionality of the problem in general, I'm not really a big fan of that approach, because I think it puts too much of our own, I don't know, yeah, and nature is not. I don't see any evidence that nature is actually playing with that kind of an algorithm for making proteins.
Thank you.
Yeah. Yeah.
It was interesting. By the way, thank you for the talk. It's great. But it was really quite interesting when you were looking at the fungus as an example, and
What?
The fungus.
Oh yeah, yeah.
And the fungus had a more. I'm going to use the term waste, because the evolution is so much older.
Yeah.
And I'm curious if you can look at that profile for different organisms that have evolved, and the proteins in those organisms, if that changes.
Yeah. So you know what's really interesting, that and I've just started to realize this, and we've written it into the paper. This text prompting business has a relationship to this. So if you notice that this prompt has left out phylogeny, right? That one has the phylogeny in it, right? So actually, the way I think about it now, there's this giant space, whatever it is, 10 to the 50 power, right? And then what you've got is evolution is like a trajectory in this space leading to the paralogs. The actual show one SH domain is one. I think what's happening here is that the prompt, this big prompt at top, is sampling more locally from the phylogenetic environment of the Saccharomyces guys or whatever. And then this one is sampling from a much deeper space of alternate solutions for the same function, right? And that's why this thing gives you this identity and that gives you that identity. It's like basically you're backing up along the phylogenetic trajectory. One way to think about it is like that. And you're accessing some space, much larger space as you go further back. And you can choose with the text prompt, in some sense, how proximal or distal you want to sample on that thing. So I guess what I'm suggesting is that we're going to put it in the paper, as I don't know anyone will understand it, but it's like there's physics and there's evolution. And I think that there's the physical constraints, which are being purified here, and then you can dial in as much phylogenetic bias as you want by putting it into the text prompt, you know?
So you're forcing the evolution within the text prompt.
Yeah, exactly. And it would be interesting to say, then, is there like a way to explore that with text prompting, right? As a kind of experiment to try to like reconstruct the phylogenetic or evolutionary trajectories, right? It's a super nascent thought, so I think there are signs and symptoms that are potentially possible with this kind of way. Yeah. Other comments or questions? Yes.
So you mentioned that like a lot of like a pulse model fitting, like they're failing because like they're neglecting the sectors.
Yeah.
Do you have like any idea, like if we go forward, if there's like any way we can like take into account like the sectors?
Yeah. So let me tell you the answer to this. So in the original paper, we made a POTS model. You make that model implicitly at temperature 1, right? And then you sample sequences at temperature 1, and nothing works. Okay, and so the interesting thing is, but if you go to low temperature sampling, like 0.6, now you suddenly get all these functional things. So what's going on here? Like you're going to lower temperature, like depressing the energy scale, right? And then you recover these constraints that you've now suppressed, right, in the thing. But of course, what comes as a problem is you enforce a lot of noise too, right? And then you end up getting, okay. So how can one correct this? So Yakov, in this paper, we just explained the problem and didn't really give the solution, except the solution is approximately like you have to do inhomogeneous regularization, right? If you regularize the whole, all the parameters with the same global L2 regularization, the statement is you will never infer these different features fairly. You can emphasize one or the other, but you never get both, right? Or whatever. So you need to do some form of inhomogeneous regularization without prior knowledge of which parameter belongs in what thing, right? And so Yakov came up with a brilliant idea of doing this sort of stochastic Boltzmann machine, the idea of like forcing the model during gradient descent to experience this same stochasticity as the input data. Like, you know, he was fond of saying in our lab, it's like apples and oranges. You're trying to make a model at the thermodynamic limit that is fitting like crap data, right? like very finitely sampled data. So if it turns out if you force the gradient descent process to experience the same stochasticity, at least in toy models, you perfectly recover both the collective modes, sectors, and contacts with the correct inference, right? So I think there's an insight there about how to do the model learning so that you more fairly treat these features of different epistatic size. So basically the stochasticity of the model becomes a hyperparameter, right? It's like an implicit regularizer that's actually now inhomogeneously regularizing all the features, right? And I think that's a cool idea. Now that needs to be fleshed out really and tested. But I think there is some idea of how maybe to fix that problem. Okay, thanks you guys for